Welcome back from the break. We hope you're able to refresh your coffee or tea to help move us right into the remaining presentation before the Q&A panel. Our next presentation will cover an integrated platform for continuous RNA nanoparticle formulation and drying by Dr. Kurt Ristroph from Purdue University. Please join me to welcome Dr. Ristroph.
All right, everybody. My name is Kurt Ristroph. I'm an assistant professor of agricultural and biological engineering at Purdue University. I'm very happy to talk to you today about our work on an integrated platform for continuous RNA nanoparticle formulation and drying. This is a grant that's funded by the FDA BAA mechanism, and we're working in collaboration with Professor Robert Prudhomme at Princeton University and Dr. Mark Castenton and his team at Saran Bioscience, which is a contract research and manufacturing organization in Oregon. So, to begin with, I'm going to be talking about the nanoparticle formulation process that we use called flash nanoprecipitation, how it works, and how we use it in particular for preparing lipid nanoparticles to encapsulate mRNA. Then, in the second part, I'll be talking about specific instances where we've used FNP for drug delivery in general, nanoencapsulation, nanoformulation, a bunch of different types of drugs from different chemical classes. Yes, I know today is mostly about continuous manufacturing for lipid nanoparticles, but we're going to talk about some work that led up to our current work on lipid nanoparticles, which is going to be described in part one. Okay, so some potential benefits of nanoparticle encapsulation are shown on this slide. So just starting and going counterclockwise from the left, one of the main benefits that you want is to improve the circulation time in the blood or the residence time in the body of a particular active ingredient that you're trying to encapsulate. So many of the biological molecules of interest in the pharmaceutical world today are what we call biopharmaceuticals, biologics. These are peptides, proteins, and nucleic acids. The body can easily recognize these as foreign agents and really quickly clear them from the blood if they're injected, for example. So in order to shield them from those processes of the body, to allow those drugs to have more time to do their work, we want to encapsulate them into some nanoparticle carrier that is small enough that it can still move around in the body, but provides a surface layer that is potentially invisible to the immune system, for example. Next on the list is targeting. So one of the things that people are trying is to coat the outside of those nanoparticle delivery vehicles with some chemistry that will allow it to target to specific tissues, specific organs, or specific cell types, for example, macrophages, other white blood cells, or tumors. The next bullet point here is co-delivery of multiple payloads. So let's say that you have two different drugs from different chemical classes that aren't very physically compatible with one another, right? You can imagine, well, many drugs are given as cocktails, where you have multiple drugs that are supposed to act together in concert to improve what we call therapeutic synergism. Okay, if the drugs are very physically different from one another, they may not be easy to dose at the same time, right? And if you want those drugs to reach the same place in the body at the same time, we and others have shown, okay, if you co-encapsulate these things into the same nanoparticle, into the same drug delivery vehicle, then you can physically package them together and get them, optimize their chances of reaching their target tissue at the same time. Over on the right-hand side of the slide, we talk about penetrating physiological mucus. This has applications for both oral drugs and drugs that are given as an injection or an inhalation, for example. Physiological mucus is a major physiological barrier. A lot of drugs get caught in that physiological mucus. That's what it's there for, is to catch foreign agents. And so if, for example, you have an infection that's underneath the mucus layer, it's very hard to get a drug down underneath that mucus layer to treat those, let's say it's bacteria, for example. So if you can package your drug in a delivery vehicle that can penetrate through that mucus layer, then you could potentially reach the site of infection better. This is all about trying to get drugs where they need to go better. The next bullet point is controlled or stimulus responsive release. So for example, for a lot of these biologic drugs, biopharmaceuticals, peptides, proteins, and nucleic acids, maybe you want them to stay inside of their delivery vehicle until they reach the place where you want them to do their work. And maybe there's something different about the place that you want them to do their work. Maybe the pH is a little bit different. Maybe the temperature is a little bit different. So if you can design a drug delivery vehicle that is temperature responsive or pH responsive or responds to some enzyme that's present at your target location, then maybe you can induce drug release sort of on demand once the drug or once the delivery vehicle gets to where it's going. It can somehow sense where it is and know to let go its payload in that location. And then the last example here is for hydrophobic drugs, for things that don't dissolve in water, we use nanoencapsulation to improve their dissolution kinetics in the gastrointestinal tract. Okay, so the technique that we're bringing to the table is flash nanoprecipitation. This was developed in Professor Prudhomme's lab over the last 20 years or so as a strategy of encapsulating very strongly hydrophobic small molecule drugs. It was originally designed for small molecule cancer drugs, which needed delivery solutions, needed formulation and delivery solutions because their solubility in water was basically nil. So the way this strategy works, like other nanoprecipitation processes, which I'll talk a little bit about, is you can take a strongly hydrophobic drug, shown here on the, shown here on the, in the yellow dots here on the left-hand side of this slide. This drug won't dissolve in water, but it can dissolve in an organic solvent. So if you dissolve this along with a amphiphilic stabilizer, that's a molecule with some hydrophobic region and some hydrophilic region, all these things will dissolve in an organic solvent. Then if you rapidly mix that organic solvent against water, then the result will be a mixed solvent environment that has too much water for the hydrophobic things to stay dissolved. They will then start to precipitate. Okay, they will be diffusing around, and as hydrophobic things find one another by diffusion, they'll stick together. Okay, and this will form your nanoparticle core when your drugs stick together, and then the amphiphilic stabilizer will adhere to the surface of that growing particle core and prevent further growth. So that's shown as a timeline on the right-hand side of the graphs, or on the right-hand side of the slide. If you do your mixing at time 0, if you get a well-mixed system very quickly, then all of this self-assembly can happen homogeneously if your system is already well-mixed by the time all of this is happening. And you can end up with what we call core shell nanoparticles that have a core consisting of the drug you were trying to encapsulate and a shell consisting of, let's say, that amphiphilic polymer on the surface. And you see there are a couple of real tricks to this. One is that the game is over after 50 to 100 milliseconds if you're making, let's say, a 100 nanometer particle. So self-assembly happens really, really quickly. All right, we're going to talk about why that's important. By the way, what you get out of this, like I said, it's a core shell nanoparticle. We know how to tune the size of the particle, and if we have a hydrophobic drug in the core, usually our assembly is so fast that the drug doesn't have time to crystallize, so we can trap it in an amorphous form, which is a benefit for us. Okay? If you don't do your mixing very fast, then over the time scale of that particle assembly, you can end up with non-homogeneous precipitation. Okay, that's undesirable. So you can see here I've used red to denote regions of the organic solvent and blue to denote regions of the water and purple to denote the mixed region. If your mixing is slower than your particle assembly, then you'll have some regions with lots of drug and lots of water and some regions with lots of polymer and lots of organic solvent. You'll have this inhomogeneous, non-homogeneous solution, and therefore your particle assembly will also be heterogeneous, and you won't end up with as reproducible of a system as if your mixing is really, really fast. So in flash nanoprecipitation, our mixers are set up, and the geometries of our mixers are set up such that when we introduce these streams into the mixers under what we call turbulent flow, so this is fast mixing, we get homogeneity on the order of about 1.5 milliseconds, which is much faster than the 50 to 100 milliseconds that it takes for our particles to assemble. So that's one of the key takeaways for flash nanoprecipitation is that you want your mixing to be faster than your particle assembly. And particle assembly isn't something you can really control the time frame of. It's going to be that 60 to 100 milliseconds, and so therefore, if you want to be in the game, you want your mixing to be really, really fast, much faster than that 50 to 60 milliseconds. We use turbulent mixing to achieve this. The other key point is that all of your compounds, your core and your stabilizer, have to precipitate on basically a similar time scale to one another. Okay? If your drug precipitates much more slowly than your polymer, then your polymer will self-assemble with itself and form these empty polymer micelles before the drug has a chance to precipitate. What that means in practice is that we need usually very hydrophobic drugs for this precipitation process. We're going to talk about an exception to that, a major exception to that, over the next couple of slides. So these are two of the main points. By the way, this is not a T mixer. This is, we call this a confined impinging jets mixer. There are two geometries for flash nanoprecipitation mixers. I'll show those in a couple of slides. So there's a cavity inside the mixer that allows that turbulent field to evolve and gives us the really good fast mixing. So it's not exactly like a T mixer like you'd get from a chromatography tubing. Okay. So for nanoparticles that we make by flash nanoprecipitation, we get a very dense layer of polymer on the surface, which is attractive for things like mucus diffusion. We can have mixtures of surface polymers as long as they're all properly amphiphilic, and we achieve independent core and shell chemistries. Okay, so if a core is hydrophobic enough to precipitate, we can trap it with a bunch of different polymer options and vice versa. Okay, and when your core material is sufficiently hydrophobic, if it has a log P, where P is the partition coefficient between octanol and water, greater than about 4.5, we're usually seeing about 100% encapsulation efficiency of that really strongly hydrophobic drug, and we can get particles that are up to, recently up to 90 weight% drug by mass, which is attractive from a cost of goods standpoint. If your delivery vehicle is mostly the drug that you're trying to deliver, then you're not paying a bunch of money for other things other than your drugs. Okay, so a couple of advantages from flash nanoprecipitation. The process is scalable. I'll talk about that more in a couple of slides. And it's very reproducible. Because your mixing is happening so fast and all of the majority of your particle assembly is happening under that well-mixed environment, we get very reproducible particle assembly, and the process can be run continuously. So one of these mixers here, for example, the only moving parts are the streams themselves. So as long as we have good pumps to pump the two fluid streams through the mixer, we can continuously operate. Challenges for F&P include, well, what if you have a drug that's not hydrophobic enough to precipitate really fast on the same time scales as your amphiphilic polymer? And if we're looking at things like global health applications, malaria or tuberculosis, or if you're looking at the agrochemical space, we need extremely low-cost excipients. Even if the only excipient we're using is the polymer stabilizer itself, we need to find very, very, very low-cost options so that the overall cost of goods of that formulation will be low. Okay, here are the two different mixing geometries that we have for flash nanoprecipitation. The first one here is called a confined impinging jets mixer, and you can see that cavity that allows the turbulent field to evolve. Here is a computational fluid dynamics calculation of where the point of mixing is the highest. So you see the point of mixing is highest right in the center of the mixer, and then lower on the outside of the mixer. This geometry requires that the two streams coming in be of almost equal flow rates so that all the mixing can happen in this cavity. In contrast, we have a 4-inlet mixer called the multi-inlet vortex mixer, we call it. This guy has 4 inlets, and particle assembly and mixing all happens in the center here. And again, you can see on the CFD graphs here that the intensity of the mixing is highest right in the center of the MIVM, which is really attractive because we want our assembly to all happen away from the walls. We don't want any of our hydrophobic materials to stick to the walls of our mixer. That could potentially lead to fouling. So here our mixing, our really good mixing and our precipitation are occurring in the center of our mixer, which is good. One of the benefits of this particular this particular 4-inlet mixer geometry is that our mixer, our streams don't have to have equal velocities like they do here. So here the two streams have to have equal flow rates, but here we have much more flexibility in those. Okay, I'm going to talk a little bit about the scalability of this continuous process now. Here are 3 different scale FNP mixers that we have in the lab. Here's the confined impinging jets mixer. It outputs 60 milliliters of particles a minute. Here's one of our MIVMs that outputs 1.5 liters a minute, and here's a larger scale MIVM that operates at outputs 5 liters of nanoparticles per minute. And for reference, the MIVM, the 5-liter MIVM here, is about 5 or 6 inches in diameter. So this whole thing, it's still a fairly small device, and there's fundamentally no limitation on us designing a even larger scale mixer if we wanted to. Usually the limiting factors are getting a good pump and the cost of goods for the materials that we're encapsulating. Okay, I'm going to go through why the process is scalable and why it contrasts a little bit to the microfluidic mixing that some people do for nanoprecipitation. Nanoprecipitation occurs when you do this mixing between something that's dissolved in an organic solvent and water to drive precipitation of it. So we can either have mixing that uses turbulent flow, like flash nanoprecipitation. The other kind of flow is laminar flow, which is the type of flow used by microfluidic devices. So turbulence, for the engineers in the audience, you're probably familiar with this. For those of you who don't know about Reynolds number, this is the Reynolds number. It's a dimensionless group that is the product of the density of a fluid, the velocity of a fluid, and the channel size that it's flowing in, divided by the viscosity of the fluid. Okay, so if you want to mix 2 fluids, let's say you want to mix a fluid, this Reynolds number is a function of the velocity and the channel size. So if you want turbulent mixing, you have to have high Reynolds numbers. Okay, so traditionally Reynolds numbers below about 8,000 are non-turbulent, and Reynolds numbers above about 8,000 are turbulent flow. Okay, so if you want turbulent flow, you have to have a high Reynolds number, which for a fixed fluid where you can't change the density or the viscosity means you have to have a high velocity or a high channel dimension. In contrast, for microfluidics, if you need a low Reynolds number, so the only knobs you can tune are velocity and channel size, you want those both to be low. Okay, so microfluidic mixing is very reproducible and there are many elegant, very beautiful science papers about using microfluidic mixing for a variety of things. We use turbulent mixing in part because of this next equation. Q here is the volumetric flow rate coming out of the mixer. So Q, like Reynolds number, is a function of the velocity and the diameter of your channels. So Q is pi v d squared over 4. So you can see that if you're using turbulent flow, you're in a Reynolds number regime where you need your velocities to be high and you need your channel sizes to be large. Okay, those two contribute straight away to a high volumetric throughput. Okay, what does that mean? That means that when you use turbulent mixing, when you use turbulent flow to drive your mixing, your process is inherently scalable. It's inherently large scale. By going up in Reynolds number, by going up in velocity and channel diameter, you're building in higher and higher throughput. And in contrast, by staying at those very low Reynolds numbers, the laminar flow mixing gives you an inherently lower throughput. Okay, so we say turbulent mixing is one, your mixing is fast, which is very attractive, and 2, your throughput is built in. Okay, and here's an example of this. You can see this is data from a collaboration that we had with a contract research organization who scaled up one of our formulations. We were making exactly the same nanoparticles in a 5 milliliter batch in the lab that they were making at the 3,000 liter scale in industry. Okay, this is a graph, by the way. This is a particle size distribution graph where we have the diameter of our particles on the x-axis and the intensity of our particle scattering on the y-axis. Okay, so you can use this flash nanoprecipitation rapid mixing setup to make lipid nanoparticles. If you want to encapsulate mRNA, for example, you load your mRNA in the anti-solvent water stream that you're using to induce precipitation of your hydrophobic lipids. So here these lipid nanoparticles are made of 4 different kinds of lipids. There is a zwitterionic phospholipid that helps to form the interface of the nanoparticle. There's cholesterol that helps the structure of the particle. There's a PEG lipid, that's a lipid that serves the role of the amphiphilic stabilizer from the previous examples. This sits at the surface and has a hydrophilic polymer on the shown here in purple. And then you have a cationizable lipid. This is a lipid that is cationic below a certain pH. And why we use this? Well, it's cationic, which we'll use to form an ion pair with the anionic phosphate groups on the backbone of your nucleic acid polymer. That's half of the molecule, that's the cationic half. The other half are these hydrophobic tails that add hydrophobicity to your anionic mRNA. So basically these two guys will come together and form an electrostatic interaction. It's like an ion pair. And the resulting ion paired complex will no longer be charged because all the charges have found a partner, an oppositely charged partner, and will have sufficient hydrophobicity to precipitate. And that hydrophobicity is coming from the hydrophobic tails on the cationizable lipid. So these guys come together and form a hydrophobic salt of mRNA that will precipitate along with your zwitterionic phospholipid, your cholesterol, and your PEG lipid. That forms your nanoparticle. And again, we want to do all of this under really fast mixing such that the electrostatic interaction and ion pairing between the cationizable lipid and the mRNA, and then the subsequent precipitation of that complex along with the precipitation of all the other hydrophobic lipids, everything is too hydrophobic in this mixed solvent environment to remain dissolved, so it all precipitates out. We want all of that to be homogeneous, and so therefore we want our mixing to be as fast as possible. We want our mixing to be much faster than the time that it takes for all of that precipitation and ion pairing to occur, which is in the time scale of, again, 60 to 100 milliseconds or so. So here on the right-hand side of this slide are 4 different particle size distributions for different lipid nanoparticles that we've made encapsulating different RNAs. One's an mRNA, one's a small interfering RNA, one has 2 different kinds of mRNA co-loaded together, which is very important for some applications. And the last one has an mRNA co-loaded with some proteins. So let's say now that we've explained at length the flash nanoprecipitation process, but that's just one step in the overall procedure for manufacturing mRNA lipid nanoparticles. Okay, so nanoprecipitation comes first. I put flash nanoprecipitation, which is Perdue's way of making using turbulent flow to drive really fast mixing, but there are other forms of nanoprecipitation. You'll hear about another very good rapid mixing process from Dr. Costa later on today, and I think from some others over the course of today. So anyway, you can sub in your favorite mixing process for this block. Then what you have coming out of that mixing process is mRNA lipid nanoparticles that are in some mixture of usually water and ethanol. Okay, the ethanol was used to dissolve all the lipids in the previous slide, and then the water was used to dissolve the mRNA. So what do you do with your effluent stream that came out of your mixer? I've shown, well, one arrow is going into tangential flow filtration or diafiltration. This is another continuous process that's used for buffer exchange. So let's say, for example, that you form your lipid nanoparticles at a pH of about 5.5, which is required for making sure that those cationic lipids that you're using to ion pair the mRNA are properly charged. That's a good pH for nanoprecipitation, for driving that ion pairing complexation, but it may not be the best pH to store your lipid nanoparticles after they've been made. So if you want to change the pH, or you want to swap out the salts that you used during precipitation for better salts for storage, you can use a process like tangential flow filtration or diafiltration. Like I said, again, this is a continuous process. It's also a very standard pharmaceutical process that's been implemented at the large scale, so we know how to do this. You can also use this to remove any residual ethanol from your particles, which is desirable from a stability standpoint and from an injectability standpoint. Okay, but you notice there's an offshoot branch from our effluent mixer, our mixer effluent, that goes into what I'm calling the analytical box. Okay, so if you're continuously making your particles, you will want to sample from your effluent, from your mixer. We say F&P is very reproducible. There are other very reproducible large-scale mixing processes, but of course with any industrial process you want to have inline sensors to tell if what you're making is in spec or not. So here are some of the analytical techniques that are employed for lipid nanoparticles. The first and sort of easiest one is dynamic light scattering, which tells you what the size of your nanoparticles is. Okay, I've shown you a couple of particle size distribution graphs over the last several slides. That's that particle size on the x-axis, particle light scattering intensity on the y-axis graph. This is where that comes from, and you can install dynamic light scattering either in line, right downstream of your mixer and before you go into TFF, or you can pull an aliquot from your mixer effluent and go check by offline DLS. Okay, and there's, I think, significant discussion today and throughout the industry and in the regulatory space about where exactly you want these different analytical techniques to be in your overall process. Do you want them all to be in line? If so, how do you make them all in line, etc. Another assay that's used is the ribogreen assay. This measures mRNA encapsulation efficiency, which we expect to change as a function of things like pH and ionic strength. So when you're thinking about what buffer you make your particles in versus what buffer you diafilter your particles into, using the ribogreen assay will help you ensure that your mRNA is still encapsulated. Then another question is whether your mRNA has degraded or not. The stability of these formulations is a significant concern to a lot of different people, and so the technique there, you can use capillary or gel electrophoresis to tell if your mRNA has remained chemically stable or if it's started to break down. After tangential flow filtration, where maybe you've put your particles in the right buffer, you've got a couple of options. So you can fill into vials and freeze right away, and that can be your final dosage form. This was the final dosage form for the Moderna and Pfizer COVID-19 lipid nanoparticle vaccines. Those were, remember, shipped around the world at low temperatures as a frozen block of ice. That's one option. You could also go from freezing into freeze drying. This is an option that a lot of people are exploring. This is also called lyophilization. This is when you use a low pressure chamber to drive sublimation of the ice in your frozen sample. Basically remove all the water in the form of ice from your sample, and it leaves you with a dry powder that you can then redisperse in water prior to injection. As an alternative to this freezing and freeze drying route, you could look at some other drying operations. And why are we looking at these drying and freezing operations? This is for storage. This is because most of these lipid nanoparticles are not going to be made and used right away. This is part of the manufacturing challenge. So let's say that you want to remove the water from your particles. There are a couple of reasons why you would want to do this. Stability, shipping, and storage, generally. So water is expensive to ship. It's heavy. We also think that water facilitates degradation of the mRNA that's encapsulated into your particles. So if you can dry these out, we expect that you'll get a much more stable powder over time, which is good. And maybe it doesn't even have to be stored at those very, very low temperatures. So that's why we're looking at other drying operations. Spray drying is one option there. Spray drying is a traditional process that's used in the food and pharmaceutical industries. We'll talk about that in a little bit. Spray freeze drying is in between freeze drying and spray drying. And then there's also an option like electrostatic spray drying, which is an alternative form of spray drying. Okay, and this will give you your final dosage form. You can take your final dosage form then and continue to do these analytical tests. If your final dosage form is a powder, you would redisperse it prior to doing some of these assessments. If your final dosage form is a frozen vial, then you would thaw it out prior to doing these, and you would use this to check stability over time. Okay. Looking at some of the other options a little bit in more detail, there are pros and cons to these different downstream processing operations that you want to think about. This is part of what we're doing in the grant, is we're trying to compare these different downstream options to one another, using as our upstream way of forming nanoparticles the flash nanoprecipitation procedure. Okay, so spray drying is continuous, it's large scale, it has medium energy intensity, it's not the most energetically expensive process there is, and it gives you a flowable powder, which is desirable for a couple of different dosage forms, like a dry powder inhaler, right? Not all mRNA lipid nanoparticles are expected to be given as an intramuscular injection in somebody's arm. That works fine for vaccines, but if you want to have, for example, something that's delivered by dry powder inhaler, you'd want a more flowable powder. Okay. Cons of spray drying: it is a process that uses elevated temperatures, and there's concern around degrading the mRNA. Spray drying is also something that's, practically speaking, it's very difficult to optimize at the lab scale because it requires a lot of material, and at very low batch sizes, you're not getting a lot of material out of the process that you can characterize. So actually, spray drying is one of those processes that's hard to scale down, and the minimum sort of lab scale that people work at, it requires a lot of lipid nanoparticles to run, and lipids are expensive, and mRNA is expensive, so it's a little bit tricky to optimize, particularly at the academic lab scale, I'll say. Spray freeze drying works at lower temperatures, which is good for the mRNA stability, and it gives you a flowable powder still, but it requires very high energy intensity, and it's a batch-wise process. Lyophilization, or freeze drying, is fairly similar. It works at very low temperatures. It's a really well-established unit operation, but it has some cons noted here. It's a batch-wise process. It has high energy intensity. The lyophilized cake that you get out isn't a very flowable powder. It's not as easy to load into something like a dry powder inhaler. And this lyophilization process does require that you freeze your particles prior to freeze drying, which is its own unit operation that brings its own challenges. Okay, so this is some of what we're doing in this grant, right, is we're looking at the overall process of lipid nanoparticle manufacture. We say, well, we've got the very good upstream unit operation figured out. We know this flash nanoprecipitation works really well. It gives you this really good mixing. It gives you very homogeneous particle assembly. It gives you really reproducible production, and it can and has been scaled up in the pharmaceutical industry. So now we want to compare several of these different downstream unit operations to try and find some of the best ones, and we're working on potentially improving some of those unit operations, optimizing them for lipid nanoparticle manufacturing. Okay, so that's the first part. In the second part, I'm just going to go quickly through a couple of examples of how we've used flash nanoprecipitation to formulate small molecules in biopharmaceuticals, and the point of these examples is to show you a couple of those downstream unit operations in action. So for example, we wanted to work with some drugs for global health indications, so malaria, tuberculosis, etc. So we had to adapt the FNP process, which I showed you on slide 2 or 3, to work with very, very low cost excipients. So for example, we swapped out the diblock copolymer stabilizer that we traditionally use in our polymer nanoparticles, which are different from lipid nanoparticles only in that there's a diblock copolymer on the surface and that the core is made up of mostly our drug rather than a mixture of lipids. Anyway, we swapped out the diblock copolymer stabilizer for another amphiphilic polymer that was significantly cheaper. This is HPMCAS is a common pharmaceutical enteric polymer that's used in tableting all the time. It turns out that that can stabilize our nanoparticles against aggregation, which is really attractive, and its cost is attractive from the global health standpoint. The other thing, another adaptation to FNP that we've done, I sort of already shown you this, but incorporating the idea of hydrophobic ion pairing. So this is how you make the lipid nanoparticles, is you take something that's hydrophilic and won't precipitate in a traditional nanoprecipitation process like this, and you make basically a hydrophobic salt out of it by ion pairing it with an oppositely charged hydrophobic molecule that can be loaded into your organic stream. And then you can add a diblock copolymer stabilizer, for example, and get your polymer nanoparticle that's almost entirely loaded with drug, which is attractive. This works really nicely for small molecules that need to be ion paired to drive precipitation, or smaller things like peptides and proteins. For RNA, you have to go all the way to a lipid nanoparticle because you need this particular mixture of lipids for good particle formation for biological activity. Okay, so here's an example of improving the bioavailability of a strongly hydrophobic drug. This is lumefantrin. It's an anti-malarial. It's extremely hydrophobic, and we wanted to make a solid oral dosage form that could be given to, say, a mother in Southeast Africa, or Sub-Saharan Africa, or Southeast Asia rather, and is given in the form of a paper sachet that can be opened and mixed with water and then fed to an infant, for example. So we couldn't make a tablet, we couldn't make a capsule, we wanted to make a powder that would improve the dissolution kinetics of this drug, because like I said, it's very strongly hydrophobic, so if you take it by mouth, it won't dissolve in the time that you have, in the time that it's in your GI tract. So we took lumefantrin and put it through our flash nanoprecipitation process using this HPMCAS low-cost polymer as the stabilizer. We got 200 nanometer particles that had 90 weight % lumefantrin by mass. We then put those through tangential flow filtration to concentrate them, and then put them through spray drying. So some of these unit operations are familiar to you now that we've talked about what downstream of nanoprecipitation may look like. So here is a graph of a particle size distribution graph of the lumefantrin nanoparticles before and after all these different unit operations, and what you'll see is these curves are all basically the same, where particle size was the same right after flash nanoprecipitation, after TFF, and then the powder that you get out of spray drying, if you add that back to water, we see that our nanoparticles redisperse back to pretty much their original size, which is really attractive. Okay, and then when you feed these to mice, or I think rats in this case, in an animal test, we saw that the bioavailability of the lumefantrin was improved 4.2-fold compared to the unprocessed drug, and that bioavailability held up after the powder was aged at 75% relative humidity and 50 degrees Celsius in a vial for a week. So this is a graph of plasma concentration of lumefantrin in the blood of the rats over time, and you see that the black and the orange, which are our nanoparticle powders, outpace the unprocessed lumefantrin quite significantly. Note that this is a logarithmic axis. Okay. As another example, we can, pivoting gears a little bit, we're going to move to flash nanoprecipitation with hydrophobic ion pairing for encapsulating. Here we're going to talk about polymyxin B, which is a peptide antibiotic that's commonly used. We wanted to encapsulate this using this hydrophobic ion pairing approach using flash nanoprecipitation, and demonstrate not only that we could encapsulate it, but then also find whether there was any improved efficacy of the drug from being encapsulated into a nanoparticle, like what I talked about on the first slide. These benefits of nanoencapsulation. Are we getting controlled release? Are we getting better circulation time, etc.? So what we found was we could encapsulate polymyxin B, whose structure is shown here. It has these 5 positively charged groups on along its sequence. We ion paired this with a very common fatty acid found primarily in olive oil, or is the primary component in olive oil rather. This is oleic acid. These will form a hydrophobic ion pair, and that hydrophobic ion pair will precipitate and can be stabilized by a diblock copolymer. We found that by changing the ratio of oleic acid to polymyxin, we could tune the release rate of polymyxin from the nanoparticles when we incubated them in a salt solution, which was really exciting. So you see we get the fastest release when we have one equivalent of oleate, and then slower and slower release as we're adding more and more of this hydrophobic counterion, which translated to really nice improved efficacy in a mouse lung infection model that we did with Jian Li at Monash. So here we're, these mice have a bacterial infection, and we're delivering either unencapsulated polymyxin B or our nanoparticles through an intratracheal route, and finding that this is the log 10 of colony forming units per lung. We find that our nanoparticles significantly reduce the number of bacteria that are in the lung over time. So this is no treatment, this is treatment with free drug, and then this is treatment with our nanoparticles. Okay. Another quick example, we can use this hydrophobic ion pairing flash nanoprecipitation combined approach to encapsulate proteins also. So here we encapsulated lysozyme, which is an enzyme with a molecular weight of about 14.4 kilodaltons. We got basically 100% encapsulation efficiency, and the exciting part here was that this lysozyme is an enzyme, and we showed that its activity was maintained after it was encapsulated and released. So making a hydrophobic ion pair out of it and precipitating it into these nanoparticles didn't affect its enzymatic activity, which was really exciting to see. Over here on the right side, we see another protein we encapsulated, ovalbumin. This is a good model antigen for making vaccines. So we got 90% encapsulation efficiency, and showed that when we dosed these nanoparticles nasally into a mouse model, we got 10-fold improvement in immunogenicity of our nanoparticles compared to free ovalbumin. And again, we think this is because we're seeing controlled release over time, and we're seeing things like the ability of our particles to diffuse through mucus, so they can reach the immune system more easily to be recognized and induce that immune response that we were going after. Last example real quick, we put, when you encapsulate molecules into these nanoparticles, you can also take advantage of the surface chemistry that you install. So here we put a targeting moiety on the end of the polymer chains that we were using to stabilize our nanoparticles. Here that we were encapsulating an anti-tubercular peptide. Tuberculosis lives inside of macrophages, so we put a mannose moiety on the end of these polymers to go after the mannose receptor that's on the surface of macrophages, thinking that, well, if the macrophage wants to uptake mannose, if we put mannose on the surface of our particles, then the macrophage will uptake the particles more, and that means that our anti-tubercular peptide can get at the tuberculosis that's inside the macrophages. And this is what we found. If you had particles that had no mannose on the surface, they were indistinguishable from the control. As you start to, if 4% of the polymer chains on the surface of your particle have mannose on the end, we get, what is that, a log and a half knockdown of our tuberculosis, and if 70% of the polymer chains on the surface of our particle have that group, we get another 3/4 of a log unit of knockdown, which was really good to see. So this is an example of a targeting story that we can do with our flash nanoprecipitation produced nanoparticles. Okay. Here's a snapshot of my lab just to close. We're looking at co-encapsulate. Well, we're very interested in all those sorts of things I've been talking about so far, and we're really excited to continue working with the FDA. We're very interested in the idea of co-encapsulating these biologics along with hydrophobic small molecules. I talked about this a little bit in the beginning with combination therapy. We're looking at how, when you do this hydrophobic ion pairing, to make hydrophobic salts out of peptides, proteins, or nucleic acids, those hydrophobic salts pack into different liquid crystalline phases in the cores of your nanoparticles, which is interesting and is the mechanism behind the different release rates that we see. So we're studying that. We're looking at unit operations for making nanoparticles at scale, like we're talking about. We have this very good flash nanoprecipitation platform for making nanoparticles, and then we want to look at things like TFF, spray drying, and other downstream ways of supporting that. And then also, I mentioned that we are working with very low cost nanoparticle formulations for example, global health indications, and also in the agrochemical space, we're trying to do nano delivery of many different biological actives for plants. I'm currently looking to hire some postdocs and graduate students. If anybody knows any good candidates, please do encourage them to contact me. None of this would have been possible without the very hardworking members, both of this overall grant team, Professor Prudhomme, Mark Cassington and his team at Saran, and then also my team here at Purdue, and our different funding agencies and partners across industry and academia. Lastly, I'll put in a plug for the Department of Agricultural and Biological Engineering at Purdue. Both our undergrad and grad programs have been ranked number 1 or 2 in the US for 13 years. We have a new building. Both the College of Agriculture and College of Engineering at Purdue are very, very highly regarded. And we sit in both, actually. We're a unique department that sits in both colleges. So lots of opportunities for undergraduate and graduate study here. Thanks very much, everybody. I'm looking forward to hearing your questions during the panel discussion and talking more about lipid nanoparticle manufacture, scale up, and processing. Thanks very much.
Thank you all for the wonderful presentations. We'll now enter into our first Q&A panel. As a reminder, if you haven't entered your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. The first group of questions will be directed to Dr. Xiaoming Xu, and here is the first question for Dr. Xu. In continuous manufacturing, how do you determine the batch record and batch size? Dr. Xu, we're not able to hear you at this time. What we're going to do is we're going to let our tech team reach out to you. We're going to move on to our next presenter, Dr. Thomas O'Connor, and then we'll check back in with you a little bit later on. So here is the question for Dr. O'Connor. If we can use CM for early phase clinical trial materials, what is the expectation for process validation?
Yeah, thank you, Ray, for the good question there. Yeah, there's the expectations are for process validation, are the same as if you were using a batch process, especially if you're in the early phase, clinical trial stages. We don't expect the process to be validated at that stage. You would have phase appropriate information. I will say that the aspect of a lot of continuous operations, because of the intensified processes, there's often opportunity maybe not have scale up or later down development, which means a lot of the data you generate during your clinical manufacturing runs may be able to be leveraged to support process validation as you get ready for commercial manufacturing. So that is an opportunity that continues can potentially provide. So thank you for that question.
Thank you for responding to that question. We've got a couple more for Dr. O'Connor, and here is the next question. How do we make proposals to be considered in ETP? Do we request this during a pre-IND meeting, or is there a separate pathway?
Yeah, this is a great question. So thank you for that question. So we can do there's 2 ways I think you can do this. So and it's spelled out in the emergent technology guidance, and you can send a proposal to the program to be admitted to the program. And so the first thing we would need to know is a little bit about the technology you're proposing and why you consider it novel, and the positive impact you expect to have on product quality. And then once accepted into the emergent technology program, if you already have a molecule identified that you're using it with, you already have a development program going on, you can submit that pre-IND or an IND amendment with your questions to the agency, and just you make sure you highlight that you've been accepted in the emerging technology program so that we can be involved and support addressing those questions. If you don't have a molecule yet identified and you're you're kind of using this maybe as a platform, looking across different potential drug candidates, but you have questions about that you could help your development program, you can submit a meeting package to the ETT directly without having an IND in-house yet, and to get that early feedback. So that's also an opportunity the program provides. So thank you.
Thank you for responding to that question. One final question for Dr. O'Connor is the following. Why do you think the early CM applications are approved faster?
Yes, thank you for that question. There's two aspects that we found. So we found the approval times are faster, and as well as the marketing time. So I'll address the approval first. So it is, I think, important to note that those early drug candidates that use the continuous manufacturing were also like a number of them were breakthrough drug candidates, had that kind of clinical impact. So I mean that drove some of it, but I also think the early engagement through the ATT was a significant factor as well, because through that mechanism there was a lot of alignment already pre-built before the application arrives. So a lot of that early engagement really sets the stage for a very I would say smooth application assessment process. And then we saw the acceleration to market. So even after approval, getting that market quicker. And I think that's related to the technology. And this idea of this potential opportunity in the scale up and simplifying tech transfer that we saw that impact there. So thank you.
Thank you for responding to that group of questions. We're going to check back in with Dr. Xiaoming Su to see if his mic is adjusted. And here's the question. We'll read that again. In continuous manufacturing, how do you determine the batch record and batch size?
Hopefully the second try works. Thanks for the question. So as Tom mentioned in his talk, that a lot of definition that's provided in the ICH Q7, it's also relevant for continuous manufacturing, especially in terms of the batch size. Generally, this either is defined by the quantity of the material, whether it's input material or the output, or can be defined based on the runtime at a fixed mass flow rate. So I would highly recommend refer to the ICH guidelines, so specifically Q7 and also Q13, as Tom mentioned. Q13 is a guidance more specific to the continuous manufacturing of drug substance and drug product. So thank you.
Thank you, Dr. Shu. We hear you loud and clear. We have a few more questions for you, and here's the next question. Switching from batch to continuous manufacturing may be great and easy for brand companies. Is there ever a case also to be made for generic companies?
Thank you. This is a great question. As we've heard from Tom's talk, that there's a lot of perceived barriers, potentially with any disruptive technologies like continuous manufacturing. So as we are seeing already with the solid oral, some of the successful examples of approved continuous manufacturing drug product, there is actually quite a benefit, and the actual perceived barrier may not be that high. For example, as Tom mentioned, time to market, and also early engagement that's possible with the FDA, which allowed facilitate the development of the product. That being said, we do recognize that developing a generic version of drug product is oftentimes different than innovator product. The margin of kind of operation is more rather thin. So I think the key would be to identify the right product to develop and to start the conversion from batch to continuous. In my opinion, so one category of the product potentially can be really a good kind of opportunity is complex formulations. So examples include what we heard today, liposomes, lipid nanoparticles, and also maybe things like formulation like long-acting injectables like polymicromicelles, PLGA microspheres. So all of those products, when the manufacturing process are often more complex and multiple steps, where the quality sometimes is hard to kind of build into the product, and the failure rate when if it's high. So this is could be an ideal candidate or the product, potentially category product to be switching from batch to continuous. And also I think from the generic standpoint, oftentimes you're looking at transferring knowledge from one product to the other product, which is an ideal example to actually utilize something like continuous, because it allows you to adopt the knowledge that you have learned from developing one continuous process and apply it to the next. So overall, I think there's a lot of potential, even in the development of the generic space, to utilizing continuous manufacturing. So thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Xu, and here's the next question. For continuous manufacturing of lipid nanoparticles, what are the areas that still need more research?
Yeah, thank you for the question. So we all probably should celebrate what we have accomplished already in the lipid nanoparticle space, especially with the success of the mRNA COVID vaccine, as we heard earlier today. But I think there are still areas that we can improve. One possible area is in the material characterization, whether it's the lipid or whether it's the cargo, which is oligonucleotide or RNAs. What are the analytical techniques that is suitable to provide us more information and better information to allow us to better control and screen the material and utilize in the production? The other, I think, is an area, it's the analytical technology that may be particularly suited for process monitoring and process controls. So this means like process analytical technologies. So this potentially can enable what we consider as real-time release, RTRT, release testing. So this does offer flexibility, but it requires analytical process that's more suitable, sensitive, and robust to the manufacturing process. And also the other area I think could be potentially useful and to explore further is in the modeling and simulation space. As you heard from Tony's talk earlier about use of CFD and MD simulations, which potentially can speed up some of the understanding process about the manufacturing process or the formation process, which in the end, in turn, may benefit the optimization of the manufacturing process, maybe implementing better control strategies. So I do think there are still areas that we can still research on, and I think that's a lot of what at UConn, at Purdue, and at Princeton, where FDA has funded multiple research, is really trying to generating some more knowledge in that space. So thank you.
Thank you for responding to that question. We do have one more question for Dr. Xu, and here's the question. What will be the challenge to scale up the manufacturing of lipid nanoparticles?
Thank you for the question. I think actually this question, I think Kurt has already talked about in his talk about the flash nanoprecipitation, about the challenges and the benefit of utilizing the continuous processes to overcome some of the scale up issues or challenges. And also, I think from the other angle to look at it, if we looking at the continuous processes, and one of the critical benefit is that the product throughput is oftentimes defined or dictated by the runtime if the process is operated at a steady state conditions. In that case, the quantity of material output is really dependent on the throughput and the runtime. Mass flow rate and the runtime. So in a way, you may have less of a challenge or issue with the need to scale up the manufacturing if the desire is to generating larger quantity of material. Instead, you could have run the process longer to produce enough material. Be considered as a batch, but I will maybe keep this as a question also for Kurt to comment on later on when he's maybe able to chime in. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in for Antonio Costa, and here's the first question for Dr. Costa. How well understood is the docetaxel doxorubicin co-loading leading to the elongated particles?
Yes, thank you, and hello everyone. So I would say, with respect to the docetaxel, what we see here is it does have a log P, so it's more of around 4.2, so it is more of a hydrophobic molecule. We can see from the cryo-TEM that there's a multifaceted structure with respect to the nanoparticle. So what this can lead to is more of a lipid domains boundary effects within within the bilayer itself. There could be potential phase separations as well. And then when this potentially leads to more of a flexible bilayer. So when we combine the doxorubicin and we have the internal environment that allows for the ammonium sulfate high salt concentration, which allows for the doxorubicin to form a crystal. This could be the cause which would lead to more of an elongated particle. So it's almost as we add the docetaxel into the particle or into the mix. This is added actually from the ethanol phase, whereas the doxorubicin is added downstream after the particles are already formed. So we're actually loading and adding at two different locations in the continuous process. So this could also potentially impact the particle formation, but most likely, it's the relationship between the hydrophobicity of the docetaxel, the interaction with the lipid, which allows for more of an elongated structure to form once the crystal, the doxorubicin crystal, forms on the inside. Thank you.
Thank you for responding to that question. And the next question for Dr. Costa is the following. Why do you measure shear rate?
Yeah, so the shear rate that's measured is actually more for the nanoparticle, the nanoflow sizer, which is the inline particle analysis unit that we used. So the shear is actually more important to understand at the boundary where the particles are actually being measured for their particle size, that they're within acceptable limits, and that's more specific to that particular instrument. So it has to be if it's at a certain range. If you're above that range, then you have other convective effects that would be influencing the particle size measurement. So we just have to always make sure that we're within that certain range.
Thank you for responding to that question. We've got a couple more questions for Dr. Costa, and here's the next question. Can you explain how to measure turbidity and what is the range of acceptance?
Yeah, so for turbidity, it's an NIR approach that was used, but it's really it's more or less looking at the sample with respect to at certain concentrations. So we're able to measure based on concentration units, which is a more of an absorbance measurement, and then there's also scattering effects. So this is measured typically in ppm. And you're looking at more of the effects of scattering on the particles, and then the combination of these two, we can understand, we can relate to the overall nanoparticle concentration and turbidity of the sample. So this is important. When developing models, we can use this for predictive models. We can understand relationships between other characteristics of the formulation and and relate this overall to the concentration. So, acceptable range would really depend on the sensor, would depend on the sensor of choice, and depend on the particles that you're measuring. So really small particles will give you much lower values, and larger particles would really increase the turbidity measurement. So ultimately understanding really the sensor and your sample is important. So there's not really a specific range that would be acceptable. Thank you.
Thank you for responding to that question. We have one more question for Dr. Costa, and here's the question. What DOE software was used for the modeling?
Yeah, so we used the JMP by SAS, and with that software, really gives you, a lot of flexibility in the different types of models that we can use, full factorial, partial factorial designs, and other complex designs, or for example, d-optimal design. So this allows you to generate the models, run the experiment, and after you run the experiments, input all the data. So really, it's easy to use, great software. There's plenty of other softwares out there that can, that can be used as well. But that's the one that we used for this work. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in for Dr. Kurt Ristroph, and here is the first question for Dr. Ristroph. Can you use flash nanoprecipitation for hydrophilic API, and what is the success rate?
Thanks very much for that question. So this hydrophobic ion pairing strategy that I talked about toward the back half of the presentation, we can use that for several classes of things that you may or may not call hydrophilic. We can use that for weakly hydrophobic small molecules or biologics that have a log P maybe in that 2 to 3 range, which aren't hydrophobic enough to precipitate by flash nanoprecipitation. But if we increase their hydrophobicity using that ion pairing approach, we can. Then we've extended the FNP plus hydrophobic ion pairing platform to very soluble biologics. Polymyxin B has a log P of -5 or -6 or something like that. So to date, we've encapsulated a couple dozen strongly hydrophilic peptides and proteins using that combined approach. And we're usually, I think the lowest encapsulation efficiency we've found with something that actually formed good particles is something like 88 or 90%. So it's high encapsulation efficiency. And what I mean by things that make good particles, if you have let's say a molecule like a dextran, it's a water-soluble compound, but it doesn't have any ionizable groups that we can take advantage of in order to ion pair and encapsulate. So if you don't have ionizable residues, you can't use that approach.
Thank you for responding to that question. We've got a few more questions that came in for Dr. Ristroph, and here's the next question. Is flash nanoprecipitation limited to producing spherical core shell solid nanoparticles, or is there flexibility to produce other shapes or other products, for example, liposomes?
That's a great question. Thank you. We haven't made liposomes with FNP because of the rate of precipitation. What we get out are these solid lipid nanoparticles or core shell polymer particles. Also, because of the chemistry of self-assembly, we almost exclusively get spherical particles. Yes. So if you want something with a very high aspect ratio, that's harder to achieve. There has been some work by Rod Priestley in chemical engineering at Princeton on using that rapid mixing process to make Janus particles that have these are spherical particles that have two faces that are composed of two different polymers that phase separate. They're both hydrophobic, so they will co-precipitate into a single particle, but then they will spontaneously phase separate. So you get something that has two hemispheres that are made of different material. But those still have an aspect ratio of 1. They're still spherical, but so yeah, generally we don't get anything other than spherical particles out of this. Thank you.
Thank you for responding to that question. The next question for Dr. Ristroph is the following. What is the role of block copolymer stabilizer? Is it not part of the final nanoparticles?
Yep, the block copolymer absorbs onto the surface of the growing nanoparticle core and prevents further aggregation. So you get that first nucleation step where the core starts to assemble. And if you didn't have any diblock copolymer stabilizer or stabilizer, there would be nothing to prevent the hydrophobic core material from continuing to aggregate and forming into a big crystal. So we can stabilize these particles either with a diblock copolymer or with what the LNPs are stabilized with is a PEG lipid, usually a DMG PEG that has a fairly small lipid group and then about a 2 kilodalton PEG group. You can also use some other smaller surfactant-like molecules. Lecithin, soybean lecithin is a zwitterionic phospholipid that can be used as the surface stabilizer. I showed HPMCAS is also amphiphilic enough to be the surface stabilizer. So that's where it ends up, is on the surface. Thanks.
Thank you for responding to that question. This will be the final question for Dr. Ristroph in this round. And here's the question. You showed that the LMN formulation showed the same size distribution after spraying, drying, and redispersion in MQ water. I assume the powder would be finally redispersed in tap water for final application. Do you have size distribution data of such a suspension as well, and what would this look like?
Well, that's a great question. So the questions about the lumefantrine formulations, when they're actually applied in sub-Saharan Africa or Southeast Asia, are they going to be mixed with milieu water? No, they'll be mixed with whatever water's available. So we don't really expect the composition of the water, as long as the water's, well, we were told by the Gates Foundation to assume that water would be potable, but small differences in ionic strength or pH shouldn't affect the redispersibility of those powders into the final dosage form. We have not tested that, but actually that's a very good idea, and I've got some contacts in Nigeria who we could potentially reach out to and try and get water to use for that. It's a good idea. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist. We've got a few more questions that came in from Dr. Xiaoming Xu. And here is the next question for Dr. Xu. Depo-Dur is an extended release liposome formulation of morphine sulfate. Are there any special specific or regulatory considerations if applying continuous manufacturing techniques to produce this type of liposomes or similar liposomes containing a controlled substance?
Thank you for the question. So what you were referring to, the Depo-Dur, is one of the multivesicular liposomes that's produced through emulsification process. So generally, the size is much larger than what we consider typical liposomes. The multivesicular liposomes generally are in the micron size range. So we have now seen the commercial, at least application of the continuous manufacturing in producing this type of multivesicular liposomes. However, we do believe there is a potential of utilizing the continuous processes, such as the one that you heard today, to generating this type of dispersion. Essentially, it's oil in water kind of dispersion colloidal system to start with when you're producing the multivesicular liposome. Instead of oil, of course, this is the organic solvent we're talking about. So there is potential, and in general, I think in terms of the feasibility, when the processes involve liquid mixing, I think there is always potential of using the continuous process to produce the particles. Now, I think still some research work is needed in order to see the commercial success. But as I mentioned in my earlier beginning introduction to the symposium, I think sometimes we need to think bigger. We need to think forward about what we hope to utilize. I think if you think about the potential benefit of the utilized continuous process to produce multiple multivesicular liposome, is that you may be able to produce more homogeneous the type of particles, which we know has been a challenge. And also you may be able to better control the internal morphology, which is a characteristic kind of feature of the multivesicular liposome. So using continuous process, we believe there are some potential benefit to further improve the consistency of the manufacturing. And internally, actually, we're working on this category product, looking at how do we kind of switching from batch processing to continuous processing for something like emulsification process. Hopefully there are more interest and there are more development in the academia and in the industry on this topic in general. But thank you.
Thank you for responding to that question. The next question for Dr. Xu is the following. What is the proper step to test size PDI zeta to be filled later for registration purposes, for example, after making the nanoparticles only, or after purification only, or after concentration, or at the stage of the processing?
So thank you for the question. So my understanding of the question perhaps is related to what's where or when it's appropriate to perform a particle size measurement on the polydispersity index, or zeta potential in order to control the product. I think the answer to that is certainly you need at the end of the kind of product manufacturing before releasing it. So this is what typically considered as a batch release test. And so you would need to set the acceptance criteria or specification for particle size or zeta potential or other quality attribute. And then you would need to develop method to properly measure and control that attribute. And however, sometimes, especially we know with some of the more complex processes, and also maybe another benefit of the continuous manufacturing is that you're actually able to measure those quality attributes in line during the production through the measurement like process analytical technology or PAT, which allow you to see the quality of the product that they're producing in real time. So you may be able to start measure intermediate quality attributes such as after liposome or lipid nanoparticle formation, or after loading, or after purification. So those I think will be critical to measure in order to build confidence for the downstream process. So because again, it's a continuous operation, so all the units are linked together. So one deviation in one process may impact the downstream process. So the ability to measure in real time those quality attributes will really give you insight into how the product is developing, whether or not it's in the following the right direction within the design space that you have come up with. So I think the answer to that question is, of course, you need to have a batch release test, but also you should start developing some of the in-process controls to allow you to better understand and better control the manufacturing process. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we've got a few more questions that came in for Dr. Antonio Costa. And here's the first question for Dr. Costa. What is the proper type of lipid selection if the API is hydrophilic or hydrophobic?
Yes, I would say there's it does not necessarily depend on the API entirely. The lipid, it's a combination. So the lipid itself is more as it's going into the bilayer. So if it's more about the structure of the bilayer that's going to form the rigidity of the bilayer, and that'll be determined by the type of lipid that's chosen. So for example, if you have or the percentage of cholesterol in the bilayer if you're using a DMPC lipid, which would make larger particles, versus DSPC. And then, with the phase transition or the transition temperature of the lipid, that could impact potential for some of the molecules. If it's hydrophobic, and the bilayer could be released and or go into another particle or cell as it's during recirculation. So there's really other impacts, I would say, than just the formation and choosing a certain lipid based on hydrophobic or hydrophilic. But if it's more of a hydrophobic particle and it's going to be loaded in the bilayer, there may be proper selection with respect to the hydrocarbon tail length, saturated versus unsaturated. If there's interaction between the lipid and the potential or the API, but I don't believe that there's a clear indication of you have to use these types of lipids for hydrophilic and these other types of lipids for hydrophobic. It's a more complex relationship. Thank you.
Thank you for responding to that question. Here's the next question for Dr. Costa. What is the method to make the LNPS?
So the method to make the LNPs. So in general, you have your lipid, which is pre-dissolved in ethanol, and then you have your mRNA or other nucleic acid that would be in the aqueous phase. We call this we're calling this aqueous phase 1 that enters our injection port, and then essentially the LNPs are formed as these two are mixed together. Some critical factors to consider would be the concentrations, the N to P ratio. So there's different charge effects with the ionizable lipid that's present, and the nucleic acid. So you have to have a certain range for N to P of the lipid and the nucleic acid. Otherwise, you may form it may be too much of a charge or too less of a charge that's present, and then this can lead to instability of the nanoparticle. So it's really the combination of the type of lipid, concentration, N to P ratios, and then also on the process side, the flow rates. Under certain conditions, the flows are required to form a more stabilized jet, and you have to reach this stabilized jet in order to form the nanoparticles. So when you combine all of these together, you can have a nice stabilized formulation. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Acosta, and here's the next question. Can you give us more details on how you calculate the in vivo release of DOCs?
Yeah, so this was a study that was worked on with NCL. So for any of the protocols, I would just point to their website where on cancer.gov. So they have listed multiple protocols. So NCL protocol PH-2. It outlines the methods that were used for in vivo characterization. Thank you.
Thank you for responding to that question. The final question for Dr. Costa is the following. For mRNA LNP stability, how long did you leave the formulations in ethanol?
Yeah, so for these for the cryo TM images that we showed, so there were two different ones. One was at the 1.7% ethanol, and then the other was at the 5% ethanol. So these were left, I'd say, at least for 2 days or so, and also during a shipment to overnight to the cryo TM facility. Once they received these particles, they then did a more of a flash freeze and stored them prior to when they put onto the scope. So it was multiple days. It wasn't just we didn't necessarily control it for hours, but that could also lead to what we see of the delamination, especially in the 5% ethanol case, and swelling. So, but that does, come or bring us back to, understanding intermediates and really understanding, how long should we do transfers if we have to do a hold time or so between processing stages, which would be more of a batch approach. If you have to do a hold time, and what conditions are required and acceptable. So it's more about understanding just the the relationship between the actual nanoparticle structure and ultimately stability of these particles. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few more questions that came in for Dr. Kurt Ristroph. And here's the first question. Is the inclusion of targeting ligands on the outside of the NPs ever a complicating factor for the nanoprecipitation process? And do you get unintended interaction between these groups and the self-assembly of the core?
Thanks very much. That's a good question. Most of our targeting work has used a relatively small chemical group on the ends of those diblock copolymers. So that hexamanose moiety, for example, is not it's not going to electrostatically interact with anything. It's not going to ion pair with anything. It's hydrophilic, so it's not going to want to precipitate into the core. If you had a targeting ligand that's instead something enormous like an entire antibody, we would anticipate some potential problems there, just because if you have a diblock copolymer with a molecular weight of something like 10,000 Daltons, if you then go adding something to it that is comparable or larger in molecular weight to that polymer, then the overall diffusion of the polymer itself is going to be significantly altered, and that could potentially affect its ability to diffuse onto and absorb onto the cores of those particles during self-assembly. So we have tended to stay towards smaller ligands for that reason, and we haven't seen major issues there. Thanks.
Thank you for responding to that question. Here's the next question for Dr. Ristroph. What is the minimum volume that can be achieved with your IgM method and the IgM overall?
Good question. The smallest batches we make in the lab, we have 500 microliters of each of the two streams. I know that there is industrial interest in scaling down that mixer to even smaller minimum batch sizes. I haven't worked directly with any of the units that I think people are developing, so I can't say more about that at this time. But yeah, we're usually looking at a minimum volume of about 1 milliliter, and there is a holdup volume inside the mixers that we use of about 100 to 200 microliters. So if we put a mill through it, we get about 800 microliters out of it. Thanks.
Thank you for responding to that question. We have one more question for Dr. Ristroph, and this will be our final question for the Q&A panel. And the question for Dr. Ristroph is the following. Can the API quality be degraded considering the high pressure involved in IgM?
That's a good question too. So the pressures are actually deceptively low. This isn't like a this isn't a T mixer. It's also not a high pressure homogenizer type unit. So the back pressures, I typically will drive the syringes, or my students will drive these syringes by hand at the lab scale, at the very small scale, and then when we go up to a syringe pump, we're not seeing significant back pressure. If the question is also about, well, you're going to put a biologic through this mixer, is it going to get sheared to bits inside the rapid mixing, the turbulent mixing? We also aren't seeing that. We've put enzymes through these, we've put RNAs through these, and we're not seeing physical degradation because of that shear. So we don't think that it's a major problem. Thanks.
Well, that's all the time we have for questions in this panel. A huge thank you to our speakers and panelists for answering numerous questions that came in from our audience. We're now in our lunch break until 12:45 p.m. Eastern. Enjoy your lunch.